Okay, thanks, Prudential. Hello, everyone. I'm Aryan. I'm a PhD student at Washington State University. And first of all, I want to say I'm very grateful for the organizers to allowing me to speak here at the seminar. I've already seen many nice work being presented, and I hope some of the stuff that I'm presenting today will be interesting to people attending. So as Prudential said, you can ask questions. I'll try to take them as soon as possible. And we can also stop in the end for more questions as well. So designing new drugs rapidly is very important to tackle novel pathogens and illnesses that affect our society. And this has become very apparent with the COVID-19 pandemic. However, most of us also know that traditional drug discovery is a time-consuming process. And sometimes it can take multiple years before a new drug comes into the market. Having said that, there is one key component of this process, which is molecular optimization, where we are essentially searching a chemical space to find certain molecules that will satisfy certain properties and constraints that we care about. For example, some drug-like property. So in this talk, I'll talk about how we can use Bayesian optimization techniques to accelerate this search process. So this part of the drug discovery process, how can we accelerate that in a resource efficient manner? So a very key consideration in my talk will be how to do things in a data efficient manner. So we'll try to place us always, most of the times, in a small data setting. So although I believe most of the people here are more interested in drug discovery application, but the search problem is much more general with many different applications, even within the field of chemical design or chemistry. For example, one other application I'm involved in is nanoporous material design. So where we are focusing on the design of this class of materials called metal organic frameworks, or MOFs, as they are commonly called, which has many sustainability use cases, including storing gases in hydrogen-powered cars, or separating carbon dioxide in power plants, etc. And they have other futuristic cool applications like converting desert air to water, which I cannot talk much about today, but you should look up on YouTube. So these applications that I just talked about, obviously formally we can describe them as optimization over combinatorial structures. So in molecule design, the space of the combinatorial structure we are talking about usually is a graph. So we are talking about graph representing as molecules. So we'll be searching over a space of graphs, and we'll be optimizing some property that we care about for the molecule. However, this is also not a standard optimization problem, primarily because of the two main challenges that arises in these applications. So first, our objective function is expensive to evaluate. And this is a key consideration throughout the talk. So each candidate evaluation will be resource intensive. For example, in molecular design or chemical design, we might need to perform a physical lab experiments. So we have to keep this in mind that each evaluation is very expensive. And second challenge is just the general hardness that comes because of the combinatorial space, the general nature of combinatorial objects. And this is why we want to find optimal combinatorial structures, but we have to keep in mind that we want to minimize the number of. Yeah, hi. Yep.
I have a question about the nanoporous material search.
Yes. Yes.
Are there ways to generate like some significant amount of data about what works and what doesn't work? So from what I know, material discovery, the actual synthesis and evaluation of a material is very slow and expensive, but you might be able to use expensive but much cheaper compared to real experiments, simulations like chemistry simulations with differential equations in order to get an approximation. So how does it look for the case of these nanoporous materials?
Yes, you are right. So exactly, the question is, how do we get some supervision about nanoporous materials? You are right. So there is a class of properties which are very expensive to calculate, but then there are these simulations which are available, I think, which you can And there is also, as far as I know from my chemistry collaborator, that a lot of these simulations can be, so there is a multi-fidelity nature to it, which means you can increase the fidelity to get higher accuracy.
Right.
So there is simulations out there. And
and these simulators are that you guys are using, are they available to other researchers, or they're like private things?
Yes, they are available on GitHub. Yes.
Okay.
I'll share some links later. Yes.
Thanks.
Okay. Yeah. So these are the two challenges. And as I said, we want to find optimal combinatorial structures, but we also want to minimize the number of function evaluations. So one popular framework and very effective framework for optimizing expensive and black box functions is Bayesian optimization. And I'll give a quick background about Bayesian optimization so that we all are on the same page before getting into the details of some of the methods that we developed. So Bayesian optimization is an iterative procedure. So where we build a statistical model which will act as a surrogate of the black box objective function or the property function that we care about. And then we will use this statistical model to intelligently or smartly select a sequence of function evaluations using what is called an acquisition function. So what we are doing here essentially is that we are replacing a lot of expensive queries that we need to do on the black box objective or the property function using these cheaper surrogate model queries or statistical model queries. And we'll be also using the uncertainty of the model to select only those queries on the property function that will be informative so that we can reach our goal or reach optimal structures very quickly. So this is a general intuition behind Bayesian optimization. And there are 2-3 main components of any Bayesian optimization framework. First is the statistical model, as I said, which will act as a surrogate of the underlying objective function. And as I just mentioned now, we need to be very careful that we need models that are aware of what they don't know. That is, we want principal uncertainty estimation. This uncertainty estimation is very critical to Bayesian optimization because we are mostly operating in a small data setting. And one gold standard, especially for small data setting, is Gaussian processes models. They provide us with very nice uncertainty estimates. And I'll be talking a lot about Gaussian process models even later in the talk. So I think one key characteristic to remember is that a Gaussian process model is characterized by the choice of a kernel. So we'll be talking about which kernels are useful in what setting a lot. So this is a key point to remember. Then the second component is an acquisition function. So an acquisition function can be intuitively thought of as a utility function. So it is giving us a utility of a point. How much utility will I gain if I evaluate this point on the objective function that I care about or the property function that I care about? And one popular example of acquisition function is expected improvement. And here the utility is defined in terms of the amount of expected improvement that we will get on the incumbent best value that we have seen in a certain number of iterations that we did. And the third component is acquisition function optimization, in which we actually optimize this acquisition function to get the next point for evaluation. So this is Bayesian optimization, the general main components of Bayesian optimization. There has been a lot of work on Bayesian optimization, and it has been around for multiple decades, but especially in machine learning for last 10-15 years, there has been a ton of work focusing a lot on continuous input spaces. There has been relatively less work on combinatorial structure, which is our setting. So I'll start with talking about what are some common challenges associated with combinatorial structures. But if there are any questions about general Bayesian optimization, I can take.
Yeah, maybe one. I'm guessing this is quite a basic question, but
Right.
Just so I understand, the acquisition function, for example, the expected improvement that you mentioned, it's
Right.
concerned with improving the best found solution so far, and not so much with improving the improving the confidence or the accuracy of the surrogate model.
Right. So it is concerned about expected improvement specifically is only concerned about that. But there are other acquisition functions which you can also take into account, the uncertainty of the optimal point. And so there is a whole series of work on different kind of acquisition functions, but expected improvement, you are right about that.
Okay, but in general, these are related with improving the solution and not improving the model.
Yes.
Okay.
And I think
Awesome.
that's where, like, I think a very common question is about active learning versus Bayesian optimization. I think you are right that in active learning, we are mostly concerned about the parameters of model parameters. Here we are concerned about the uncertainty of the optimal point or the location of the optimal point. Yeah.
Clear. Thank you.
Yeah. So in Bayesian optimization over combinatorial structures, it has its own challenges. As I said, the first one among them is the challenge of defining effective surrogate model, or the challenge of modeling these input structures. And the second one is obviously solving this acquisition function optimization, which is going to be computationally hard in most of the cases. And more importantly, these challenges are related. As you can see, acquisition function is parameterized by the model. So the complexity of solving this problem is going to be proportional to the flexibility of the model that you have. Probably for molecular design, the acquisition function optimization problem is not that interesting, because in a lot of settings, we have a fixed data set and you can iterate over the data set to optimize the acquisition function. But in other settings, it is very critical. So to address these challenges, we have been working on some methods for past 2-3 years, and this is a list of our papers. Please read them, and you can definitely contact me if you want to discuss any one of them. But however, in today's talk, I'll focus on two main couple of works. So first one is a recent NeurIPS paper, where we combine the power of data-driven learned representations with hand-designed features to address the modeling part of modeling challenge that I just talked about. And the other one is the AAAI paper, where we study one particular class of models, and then we come up with some scalable solution. I'll talk about what I mean by scalable later when I come to that part, and also tractable acquisition function optimization. So I'll start with the first part now. So the first part is this recent idea that we were working on, ladder, where we are combining strengths of both kernels that are defined over combinatorial spaces or combinatorial structures, and the power of deep generative models for efficient combinatorial view. So efficient here is the sample efficiency. So again, we're talking about minimizing the number of function evaluations. And I will show how we can combine the power of data-driven representations and hand-designed features to get better surrogate model accuracy, and in turn, improve the BO performance. So this part of the talk is focused more on the surrogate model, combining these two. Okay, so let's think about a very natural way to perform Bayesian optimization over discrete spaces. I might be interchangeably using combinatorial, discrete, and structured, but they are referring to the same thing. So I'm sorry if that is confusing. So what is the natural way to perform Bayesian optimization over discrete spaces? Given that we know there has been a large body of work on Bayesian optimization over continuous space. So a very natural way is that we convert our discrete space into a continuous space and then do BO over that continuous space. So how can we do that? One very popular way is by training a deep generative model using a large set of unsupervised structures. So we do know that supervision is at premium here. We cannot get a lot of supervised data. But in a lot of settings, especially in molecular design, we are given a set of molecular structures, which we can leverage, because although a molecule is represented as a graph, but it's not any generic graph out there. It has certain properties. And that is why even hand-designed features are very useful, because there are certain special classes of graphs. So we can use the recent power of representation learning that we have seen to generate these new continuous representations and do our Bayesian optimization there. So one such example of a deep generative model is an autoencoder style model. So here what we do is we encode points that are in our original combinatorial or input space. So we are encoding a molecule through an encoder, which is usually represented by a deep neural network, into a continuous latent space. And then we will try to learn the distribution over the input space by approximately trying to reconstruct the input. So the latent space point will be again decoded through another deep neural network in most of the cases to the original input point. And then we will try to match the decoded point with the original input point. So this is the general intuitive idea of how this model works. And once we have this model, we can just perform Bayesian optimization in this learned latent space and leverage all the BO advances over continuous spaces. So this is what is called as latent space Bayesian optimization. Right? So now let me describe in terms of the components that I just talked about. So we will, in latent space Bayesian optimization, we will define a surrogate model, let's say a GP Gaussian process model on the latent space. So you could use any continuous kernel. And then this is the critical part. So the point that is suggested by acquisition function optimization first has to be decoded through the decoder network. And then that actual input structure, the molecule that you get after decoding, will be evaluated on the objective function. And the surrogate model only knows about, in the standard latent space setting, the surrogate model will get to know about the latent space point and the corresponding objective function evaluation. So you can see there could be some challenges with this approach. There could be some limitations. So first one is, this approach is not explicitly incorporating information about the decoded structures. So the surrogate model only knows about the latent space point and the corresponding objective function evaluation. It doesn't know about the decoded molecule or the structure corresponding to that latent space point. So by that, we are missing out on some important inductive bias, we call it in machine learning, but we are missing out on some important information, you can see, which might be useful in modeling, which can help us in modeling. Because we know that these structures have useful information. So for example, when we compute these Morgan fingerprints, we are using, it's some sort of a substructure counting, right? So that has shown to be very useful in predictions as well, in predictive power. It has shown to be having good predictive power. So we are losing out on that information, which we can incorporate in our surrogate model and get better accuracy. And the second limitation would be that the surrogate model, especially this is very specific to the small data setting, may not be able to generalize well, because again, we have small data setting and the latent space dimensions might be large. So these are the two limitations that might come across. To address these two limitations, in this paper, we proposed an algorithm titled ladder. So the ladder name was because we were trying to think that this is a ladder connecting the combinatorial space with the continuous space. And the key idea here is that we will improve the surrogate model with a new kernel. So as I mentioned earlier, that Gaussian process. So one thing, if you want to remember about Gaussian process in this talk, is that you need to specify is the kernel function. So we are talking about a new kernel function that will integrate both the learned representation, the latent space representation that we were already doing, but it will also integrate the rich structural information that we are getting from the decoded structures. So we will integrate that information as well. And this approach will have this, I'll talk about the key. So the structured couple kernel I described now, but this has two main advantages. One, it is a generic approach, which means we can just leverage all the complementary strengths of deep generative models in structured kernels. So structured kernels, again, I'm referring to kernels that are defined over combinatorial space. So for example, in our setting, kernels that are defined over molecules or graphs. And so because it's being generic, any advances that you make in deep generative models or any new kernel that we come up with, we can just use them in a plug and play manner. So that's the generic part of it. And another important part that you will see that we'll still be performing acquisition functions. So one critical component earlier was that because we were doing BO in the latent space only, we were doing this acquisition function optimization over continuous spaces. So we'll still be doing over continuous space, which is presumably much easier than, let's say, a combinatorial search or a discrete space search. So these are the two advantages. Now I'll talk about the two main components. So the main component of that is the structured coupled kernel. So the intuitive idea here is that this kernel is going to combine two different classes of kernels. One is the kernel that is defined over the latent space, which we are already doing in the standard setting. This could be very popular ones like Matern kernel, Gaussian kernel, etc., etc. And then we are also going to use these kernels which are defined over the structured space or the combinatorial space. We refer to them as structured kernels. This could be, for example, fingerprint kernels over molecules. So you compute the fingerprint of one molecule and the other, and the dot product between them will be a kernel value. Or it could be subsequence kernels for strings or random walk kernels for graphs. So we are trying to leverage all these advances. So kernel methods for these structured spaces has been around. There has been a lot of work for 20-25 years. So we are also trying to leverage that advances along with the recent advances in learned representations. So the key idea, okay, before going into the technical details, so intuitively you can think of it this way. So when we had only the continuous kernel, let's say we have collected some training data and then we have this. So around the training data, we assume that it is going to be good enough. But as we move away from the training data, this new structured kernel, the coupling, we are hoping that it will help us in getting better extrapolation as you are moving little away. So these terms I'm using very loosely, but now with the technical description, you will see much more clearly. So this is the general intuition here. And so how we are doing is, we are doing this by extrapolating eigenfunctions of the latent space kernel with, again, eigenfunctions or basis functions from the structured kernel. So this is the expression. I will try to decode this expression. So again, notice that this kernel, this new structured coupled kernel, denoted by C, is defined over Z and Z prime. So which means Z and Z prime are points in the latent space. So still it is defined only on the latent space. So as I mentioned, that equation function optimization can still be done in the continuous space. And the expression here is basically nothing but a generalization of the popular Nystrom method. So Nystrom method is very popular in machine learning for scaling up kernel machines on large data sets. But in principle, it's an idea. It's the general idea is to extrapolate eigenfunctions. So here, what we are seeing is Kz, as I said, will be the kernel value between the points which are decoded. So you decode the structure corresponding to Z and the structures corresponding to training data that you have collected. So K subscript Z is the kernel, that kernel vector. And the capital K is the kernel matrix between only the training points decoded. And L is the kernel matrix on the latent space. So the technical idea is this generalized Nystrom, where we are trying to extrapolate these eigenfunctions in a way that this coupling will help us in our surrogate model performance. And we actually saw it mostly empirically that we get better surrogate model performance using this kernel compared to the standard latent space kernel. So this is the idea behind the structured coupled kernel. Now I'll talk about some experimental results. So we evaluated ladder on two commonly used real-world benchmarks. One is the arithmetic expression task, and the second one is the chemical design task. So let me give a quick introduction about the experimental setup. So in the arithmetic expression task, we have a space of single variable expressions which are generated by some grammar. So these are basically strings, and we want to find an expression which is closest to certain given target expression. And the second one is the chemical design task, where we use the popular zinc data set and optimize the water octanol coefficient property or the log P property. It turns out later we got to know that log P is not the best, especially in poster sessions and all. When I attended, people said that log P might not be the best property. So we are working on other objectives as well. So you could see that to instantiate ladder, we need to define two things. One is the deep generative, whichever deep generative model we use, and the specific kernel over the combinatorial space. So for the arithmetic expression task, we used grammar VAE. And this again, as I said, you can just plug and play any good deep generative model that is out there. So any advances can be leveraged here, and use the subsequence kernel because it's a string space. For the chemical design space, we use the junction tree VAE and the fingerprint kernel. So basically compute the Morgan fingerprints and the kernel using that. And as you can see here, what we are showing here is the overall BO performance, and x-axis is the number of function evaluations. So as I said, we are focusing on a very small data setting. And we saw that ladder kind of outperforms the standard latent space BO. Everything else is kept the same. So acquisition function is kept the same. It's the expected improvement acquisition function. The acquisition function optimization is kept the same. In fact, we use two different variants of the standard latent space to see all the possibilities, if we are not missing out on just acquisition function optimization. So this was the comparison with respect to the standard latent space BO. And then we also compared with other state-of-the-art model-based optimization. We saw that ladder is competitive here as well. In the chemical design task, it is not outperforming, but we tried to tune all the baselines carefully. Yeah, I couldn't you go ahead.
You do have a few questions in the chat.
Okay, so let me see. Yeah. To already be structured with respect. Okay, so one question is, why not force the latent space to already be structured with respect to fingerprint during the training of the generative model? If I'm guessing, if I'm understanding it right, please correct me if I'm not. There is a lot of work around that. So this idea of you doing BO in the latent space came around like first you could say, I hope I'm not missing out on any other work, but generally came around 4 or 5 years ago. And then people were doing standard, but since then people have tried to come up with these different variants of deep generative models that are tailored for Bayesian or any sort of optimization. So design. Earlier because these models were earlier, the idea behind training these deep generative models were not necessarily for design or any Bayesian optimization in the latent space. So they were kind of disconnected. But now people have started thinking about these issues much more carefully. And even so, people have also started doing things like, okay, you optimize for a bit, then you retrain your model. So these kind of ideas are out there, and I can definitely refer to other related work in this space. Yeah. So this is the first question. I hope that is clear. The second one is, what information is contained in the continuous latent space codes that aren't contained in the fingerprints? Yes, that's a good question. So ideally, if we have the best model available, ideally we expect the training of these autoencoder models to capture a lot of information, but that's not always the case. And we have seen that they fall on a lot of, they remain behind on a lot of aspects. So I think as the power of these models grow and we have larger and larger data sets, obviously their representation power will grow and they will capture a lot more. But currently, I think they are not capturing everything as such. So some information is definitely lost, and characterizing it exactly in quantitative terms it might not be exactly clear to me right now, but definitely, as we saw in our experiments as well, that using these structured kernels, so the idea behind most of these structured kernels is, if I boil it down to bare bones, is counting substructures. So even in strings, you are counting substructures, graphs, you are counting subgraphs. So that kind of property I think that there is some speciality to that, because that's what we saw in our experiments as well. And I think that's what is coming across when you couple this new kernel. So that's the second. I hope that answers as well. Should I go ahead? So this was, we also, there are a lot of other results in the paper. I highly encourage everyone to see if, so we also saw that we did ablation experiments where we saw, where we compared only the surrogate model, the standard surrogate model, and the new surrogate model with the structured coupled kernel. And we found that the new surrogate model has better regression properties. So as I mentioned, that everything else in this experimental setup is kept the same. The acquisition function part or the acquisition function optimization part is the surrogate model, which is different. And we saw it in the ablation experiments as well. So for future work, again, as I said, one critical real-world application we are looking at is this nanoporous material design, where we have already done standard Bayesian optimization. So this is one recent work. Let me see another. Okay, thank you. So where we did standard Bayesian optimization on a given, this is a data set available of 70,000 coughs. These are again siblings of MOFs. So I hope I'm saying it right. So among the 70,000 coughs, we could find the optimal solution in 100 evaluation, but this is continuous feature space. So we could do standard Bayesian optimization. I think next we are looking at extending ladder to more challenges in this domain. So this was the first part of the talk. I'll move to the second part. If someone has questions, I can take. Otherwise, I'll move to the second part then. Okay. So in the second part, I'll talk about AAAI 2021 paper on Mercer features for efficient combinatorial BO. Again, efficiency here is the sample efficiency. I can go over it quickly, Prudential, if we have time. How much time do we have?
I think we have 10 more minutes for the.
Okay. So yeah, first I'll go through the intuition and the main big picture, and then we can see how much. So what is the key idea behind this approach? So we did something, we basically in this paper, we constructed what are called as explicit feature maps for diffusion kernels over discrete spaces. So let me unpack that. So as I mentioned earlier, Gaussian processes are characterized by the choice of kernel. So here we are talking about diffusion kernels, and the setting here is not graphs per se, but binary sequences. So these kernels have been shown to be very useful on binary sequences. And so what the idea behind what does a kernel do? So a kernel function basically takes a point from an input space and it projects it into a feature space. I think if I'm sorry if I'm not being, but if people have taken machine learning 101, I think it comes across very early in SVMs, where we see this picture of going from the input space to a curved space where we could do the linear things again. So that's the intuition, and that is the right intuition as well. So for example, for Gaussian or the famous Matern kernel, the feature space is infinite dimensional. So in this paper, we could try to come up with explicit expressions for these features for the diffusion kernel. And we call them Mercer features. And you might ask, what does it help with? So what it does, it gives a closed form expression with which we can sample a function from the GP posterior. So another interpretation of Gaussian processes is that they are Bayesian models. So you can think of them as distribution over these property functions. So let's say in the molecule design case, if we have these molecule property functions, so GPs, one way of thinking about them is distribution over those functions. So when we don't have any data, we will consider a lot of the possibility of functions is very large. And this possibility of which class of functions is possible is dictated by the kernel you choose. So again, so over time, our posterior is updated, which means we are closing we are trying to close on the right function, right property function as we get more data. So a very common step in a lot of acquisition functions in Bayesian optimization, including Thompson sampling, And these entropy, there are entropy-based methods where I think Cass mentioned like uncertainty-based methods, which we care about uncertainty. A common step is that you sample a function from the GP posterior. As I said, it's a distribution of our functions, so you can sample one, and then you optimize it. And this is a common step that keeps coming across in a lot of these acquisition functions. So using this closed form expression, you can do this. So this was the key idea behind this paper. So in some sense, we thought of it as a bridge between continuous spaces and discrete spaces. Discrete here, again, I'm mentioning binary sequences mostly, but they are also very general. That using this Mercer features, you can sample those functions in a closed form. It's an approximate procedure, but still getting those closed form is nice. And you might also ask, how is this relevant to molecule or drug design? So as I said, so there are two key desiderata that we worked on in this paper, which I also feel is relevant to molecule design. So the first one is some of these acquisition functions like Thompson sampling, for example. So the idea behind Thompson sampling is this. You sample a function from the GP posterior, optimize it, and the optimized solution is the next evaluation that you want to do. So I imagine you can see it very intuitively. If we can do this sampling in a parallel way, we can do multiple optimization in parallel, and all this set of optimized solutions will be our next batch. So these acquisition functions have a very easy parallelizable setting. So the setting where we want to evaluate more than one point in a single batch, but still minimizing the total number of rounds. So each round has more than one evaluation. So I think this is also, I believe, is very relevant here, because this allows us to use our parallel resources more efficiently. And the second thing is that a lot of multi-objective, so most of what I've talked till about is single objective setting where you have single property. But obviously, we do not care about single properties. There are a lot of other properties. The most natural will be a multi-objective setting for Bayesian optimization. And the acquisition functions that we use there, a lot of the state-of-the-art ones are these entropy-based ones. There, again, you can use the Mercer features to sample the function. So this is the general big picture about this paper.
I have a question. Is the reason for Thompson sampling that it makes the candidates in the batch diverse because they're all sampled from different functions? Is that
right? Right, exactly. So I think that's a very good point, because in fact, I'll show some results later. Thompson sampling has this nice property that the solutions that you get are much more diverse. It's a simple idea, but the diversity of the solutions is a really critical thing which comes across. Yeah.
Okay, so you sample a function and then you optimize it, or some amount of optimization. Right. That gives you a candidate, and you do it in parallel for all the samples of the GP.
Exactly. Yes.
Function. Okay.
Yeah. So that's the general idea. Probably I should go over. Okay, I'll quickly go over the technical part, but then I'll spend more time on the experimental section where we had one interesting application. So as I said, this paper was mostly focused on the binary spaces, but they are still quite general. So again, you require some kernel again over the binary spaces, and we used what are called as diffusion kernels. So here, when I'm talking about, okay, let me see in the chat. Seems like, okay, I'll come to the ladder question in the end, if that is okay. Sorry for, I think, Tony, sorry, I'll come to it later. So again, here, when I talk about graph representation, I'm talking about a single graph. So earlier we were talking about a space of graphs. Here we are trying to represent our entire binary space of sequences as a single graph, and the kernel will be defined on the nodes or the vertices of the graphs. So this is the expression of the kernel where L is the Laplacian. So Laplacian is a common spectral property of a graph. And then we require a graph representation. One paper in 2019, Combo came up with this graph representation where we represent the entire binary sequence on the nodes of the graph. So it will be exponentially sized graphs. But one nice property of this graph is that you can split it into graph Cartesian product over individual subgraphs. And we will use this property. We use this property to come up with those closed form expression for the feature maps. So this is the expression, like we are talking about, this is the kernel. We have written this in the explicit form. So this is what is called, famously called as Mercer theorem. I'm sorry if this is too technical jargon, but I am happy to decode it later. So basically what we are doing is we are representing it in terms of the eigenvalues and the eigenvectors. And because, as I said, the graph will be exponentially sized, because the exponentially sized combinatorial space, we have an exponentially sized terms. And the key step was how to come up with the closed form expression for these eigenvectors and eigenvalues. And that's what we did by using these three graph properties. I think I should move over to the experiments quickly, but these are three commonly known graph properties we exploited. And then we can compute the eigenvalues and the eigenvector in this closed form. And this is the closed form that you get. So obviously, the feature maps are still, the entire size of the feature map is still 2 to the power n, like exponentially sized. But as is common, we took a truncated feature map, and that showed very good performance. And also we could get tractable acquisition function optimization using Thompson sampling. So this was the idea about this paper. As I said, it allows sampling functions from GP posterior. I didn't talk about the acquisition function a lot here, but a major part of this paper was also talking about how can we get these tractable acquisition function optimization using efficient, these submodular relaxation solvers. But probably this is not very relevant to molecular design. I'm happy to discuss it as well later. So this is one of the application among other applications that we benchmark this paper, this idea on, which is of biological sequence design. Again, here we are designing biological sequences like DNAs and proteins. And the desiderator here, as I mentioned earlier, might be something which is also applicable to molecules and drug design. So the first one is, which I think just was talked about, that we want this batch. So in each iteration, we want to evaluate not a single point, but a batch of sequences. And we want to care about the diversity of these sequences to account for the fact that many of them will fail on other downstream objective. Although we are optimizing one objective, but when we use them in the real world on some other objective, they will certainly fail. So this diversity is usually very helpful. And we also want to get better real-time acceleration. I think this is a much more, this comes across naturally with Thompson sampling, as you will see. So this was one of the benchmark. It is available in these two papers. So the property was, we were trying to maximize this binding activity between length 8 DNA sequences. And we compared, so MerCBO we instantiated with Thompson sampling, the parallel version. And we also compared with the parallel expected improvement version. And we compared it on two metrics. First is the best objective value as a function of the wall clock time. So this is not the number of iterations, but the real wall clock time, and the diversity of the sequences, which we measure in terms of the Hamming distance, because these are categorical sequences. So we found that although Thompson sampling has comparable performance in terms of the objective value, but the real-time acceleration is very high. Like we can, because of this very fast parallelizability. So EI has this dependence in parallelizability, like you have to depend on the previous member of the batch to do this in parallel. So this real-time acceleration comes very nicely. And we found that Thompson sampling has better performance as we increase the batch size. And in this setting, we believe that is because of this part, the diversity part, as I mentioned. So as you can see, Thompson sampling gave us much diverse, very compared to EI. So the sequences were, the diversity was much higher compared to EI when we did Thompson sampling. So this was that. So this was the second paper. And so I think this is probably the ender part of the talk. I can take some questions now. But today I discussed basically two examples of how machine learning, and especially Bayesian optimization, I believe, has significant potential in scientific discovery. And there are a lot of open challenges still in this talk, and I'll be happy for anyone who wants to discuss. And there are a lot of open challenges for the community to work on. I also want to say, obviously, this work is not possible without the help of my advisor, Jana Dopa, and Professor Corey at Oregon State, and my lab mate, Serene. I also want to say that probably because of the time, I have not done any justice to related work in this area, and I'm sorry for that. But definitely a lot more references, and you can find a lot more detailed content. We are doing an AAAI tutorial soon, so please find it there. I'm sorry for everyone, but I'm pretty sure there is a lot of good work out there other than this work. So definitely look out that as well. And all the data and the code is available. You can go to my website. It's also available on GitHub, and I can take any questions on email, and we can also take some questions here. Yeah, thank you. Thank you for listening.
Thank you so much, Aryan. Great talk. Very much appreciated. Maybe you can start with, we can start with the question from Tony in the chat.
Yeah. So let's see. So this question is, it seems like the effectiveness of ladder relies on the effectiveness of the structured kernel you are using. Do different kernels within a domain produce different results? Yeah, thank you. That's a very interesting question, and you are right. So the choice of kernel matters a lot. And I think that's a very general statement that we know from in general settings as well, that we have to select a certain kernel, the right kernel for our setting. There are certain considerations that you can go with. Theoretically, there are some theoretical notions. There are what is called as universal kernels, which are known to have properties that they can capture any function defined on a certain space. But even in terms of practical notions, there are some considerations we can take into account, especially this trade-off between computational time and accuracy. So there are a lot of, for example, the setting of graphs. There is a huge literature on graph kernels. And there are certain kernels which, again, can give you good accuracy, but might take you a lot of time. But there are other kernels which might give you less accuracy, but they're quite faster. So these are the considerations that you definitely have to take into account while picking up the structured kernel. And definitely, it's an important consideration, design choice in the framework. Yes. Yeah. Okay. Thank you.
Cool. I have one question. When you were talking about MerCBO, you were saying it could be useful for multi-objective optimization.
Right. Right.
But you didn't actually explain how.
Yeah. So what I mentioned there was that we go back. So what I was trying to say is that because the state of the art acquisition functions in BO are these entropy based methods. One popular one is max value entropy search. That's like the state of the art in all the different settings of Bayesian optimization. Like you can construct different variants like single objective setting, multiple objective setting, or multi-fidelity setting. So these entropy based methods, they are a little expensive to compute, but they are the state of the art. And in all of these methods, the key step is this sampling. One of the key steps is this sampling from the GP posterior. So what I was trying to say is that because this idea allows sampling on the discrete spaces, so that's why, and you can use those entropy-based methods very easily. So that's why you can also use this multi-objective setting. So that was the connection there. We didn't do actual multi-objective experiments in this one, but again, even in the multi-objective acquisition variant, you will sample from the GP posterior. So that sampling part is common. Yeah.
Okay, so I guess the goal here is to sample from the GP and optimize one objective at a time and evaluate that in parallel and gather the information about.
Yes.
Okay.
Yes.
I see.
Yes,
Okay.
you're
Cool.
right. So combining that information. Yes. Yeah.
Okay. Nice.
Okay. So let me see in the chat. Okay. Yeah. Thank you for listening. Yeah.
Any other question?
I have one, but it might be a bit obvious.
Yeah, that's okay.
So you mentioned that an important property of MerCBO is the parallelization,
Yes.
which leads to real-time speedups.
Right. Right.
But for example, in the case where we're doing molecular optimization, actually doing an iteration, like the bottleneck in doing an optimization iteration would be the experiment, right? Like the wet lab
Right.
experiment or whatever.
Right. Right.
So to what maybe I misunderstood something, but I'm not entirely sure. In that setting, is the real-time speedup still a plus of this method, or is it less relevant in those scenarios?
Yes, I think that's a very good point. I think I would also say, if I'm thinking about it, yes, the real time in most of the settings, I think the real time evaluation will be much more expensive. So this acceleration might not be that applicable, but I think this idea, because this was more of a methods paper, and we also talk about other settings in machine learning. So there are settings where you could just do simulations which might be faster enough, but they have premium. So the premium might not be only on the time, but it also could be on the cost. Things could be faster to do, but they could be expensive in terms of, let's say, some other resource. So in those settings, that is useful, but probably you are right, like in molecular design, especially if you're doing a lab experiment, probably the bottleneck is always going to be the evaluation. And I think that's the central part that we have to be aware of in most of the cases, that it is the bottleneck is always the evaluation. Yeah, right. Yeah. But there are always some settings where there could be some other resource that we are utilizing.
Yeah. Interesting. Thanks.
I think I have a comment and no question. One thing I found really interesting about the talk, like especially the ladder method, is this idea of combining the kernel and the discrete space and the continuous one. Often when you work with chemists on a real life discovery in a lab project, it's quite hard to integrate the intuition in the optimization process. But this method actually allows us to incorporate that, right? Because like the discrete kernel is something that can be given by your group of chemists that you are working with. And that's a lot of good knowledge to incorporate. So yeah, I think this kind of method will be really, really useful in real life setting. So thank you for that.
Yeah, thank you. Thank you, Prudential. I think you're right. Thanks for the nice words. I think that was the intuition and the motivation behind the work. Like there is a lot of work. I mean, all of us are seeing that the advances because of representation learning in like deep neural networks and all are very powerful. And with more and more data, they are going to be very like more and more powerful. But there is also a lot of work, as you said, which has been existing on designing these hand design features, and especially for domains, it's very useful because I'm pretty sure a lot of chemists have spent a lot of time coming up with these features, and other domains as well. People have spent a lot of times in coming up with the right features, right hand designed kernels, and it's always nice to complement these two approaches if it is possible. So that was the motivation as well.